Ladies and gentlemen, let me please introduce our today's host, our today's speaker. I will spend a few words about him. It's Viktor Baranov, Dr. Viktor Baranov, and it's my pleasure to welcome him here. He started his scientific career in Ukraine by studying bachelor and master degree in Kharkov. And after that, at some 2012, he finished. He worked a little bit. I didn't study.
And then I didn't read.
Until 2014, when he was granted a scholarship, Marie Curie scholarship, and it enabled him to move to Leibniz Institute of Freshwater Ecology and Inland Fisheries in Germany, Berlin. And since then he started his PhD, and during his PhD, which he finished in 2017, he was awarded as the best student talk in the Congress of World of Bioturbation 4th Conference, and also the best talk at the 20th International Chironomid Symposium in Trento, in Italy. And also,
prosím vás, smrtěte tam za mě, fakt se nedá, už já tady mám problémy rozumět, když vy tam vzadu se jakkoliv kecáváte. Kdo to nechce poslouchat, vypadne ven.
for the students to be quiet. And during this period, there is a long list of items which say that this guy acted as a guest researcher, in, for example, in Gdańsk, in Bergen, in Russia, in Petersburg, and also in United Kingdom. So many positions until the end of the PhD. And since then, he worked shortly in Senckenberg Museum, and now he moved to Munich and got the position of postdoc at Ludwig Maximilian University and the Department of Zoology there. And they are trying to discover the origin of holometabolan insects. But now his talk will be about the PhD work on bioturbation, and also I would like to invite you to a hydrobiological seminar, because Viktor also is interested in paleo insects. So at our hydrobiological seminar, Viktor will have also a talk about some paleontology and chironomid and related insects. So please, your turn now.
Thank you for your very kind introduction, Vid. It's my pleasure to be here, ladies and gentlemen. So just a quick explanation. So I'm working both as a freshwater biologist and insect paleontologist. I have a multiple interests. My PhD was about insects as a geological force, how the insect activity in the freshwater environments is altering the carbon and oxygen fluxes, and that's what I'm going to talk about today. So title of my talk is Taking a Deep Breath, because insects are actually very important drivers of the respiration in the aquatic environment. So that's some items that Vid already mentioned, so I would skip it and I would go right straight into the game. So the process through which insects are influencing all these magical ecosystem functions like carbon sequestration, respiration, cycling of the nutrients is called bioturbation. The bioturbation is a very interesting process. It's originating at the very early of the Phanerozoic, at the start of the Cambrian period. And many scientists do believe that actually bioturbation was a reason which made the Cambrian explosion of biodiversity possible, because animals started burrowing into the sediment and caused the water mixing, which broke the long-standing ocean stratification, allowing oxygen to move to the benthic layers of the Cambrian seas, which allowed larger animals to grow into the benthic environments when oxygen became available, and that has facilitated the skeletal revolution as one of the factors, among with the cooling and oxidation of the iron in the sideroid basins and so on. But It's not about Cambrian explosion, it's about bioturbation. So bioturbation is defined as an organism-assisted movements of sediment particles and fluids within the sediment matrix. It could be terrestrial sediment, it could be aquatic sediment. The first person who made a note of bioturbation was actually no one else as a Charles Darwin himself. It was one of his last ideas. There is even a paper called Bioturbation, taking a second look at the Darwin's last idea. So in his garden in the city of Down in UK, he was placing a small pieces of chalk in his garden, and he noted that these chalk pieces are disappearing because earthworms are burying them. He studied the burial of the chalk and leaf litter for about 15 years, which is a real luxury. You know, we are normally not getting experiment funded for 15 years, and it helps when it's just the chalk pieces in your garden. And he wrote his last book called On Origin of Vegetable Molds by the assistance of the earthworms, where he noted that this process is actually crucial for formation of the soils and agriculture. But unfortunately, since this work was published a year after his death, it never got a prominence it's actually deserved. So bioturbation is consistent of two major parts: sediment reworking, which is the moving of the solid particles, and ventilation, which is the moving of the gases and liquids through the sediment. Here's a number of examples of bioturbators, like this vat worm, Arenicola marina, also known as a common sandworm. And here you see the bioturbated littoral of the North Sea at the German reservoir Wattenmeer. And this is a star-nosed mole from North America. It's one of the cutest bioturbator you can get across the board. It's also bioturbating. Also, large animals could be bioturbators. Alligators are bioturbators because they dig in the holes and ponds in the Florida and Everglades. Also, elephants are bioturbators because they build in the drinking ponds in the African savannas. But I'm not here to talk about elephants. I'm here to talk about probably one of the most important freshwater bioturbators, bloodworms or chironomid larvae. So bloodworms or chironomids are extremely common animals. There are about 6,500 of modern species, and they existed for the last 210 million years, and they are the most abundant aquatic animals in most of the ecosystems, occurring from the Antarctic Peninsula to Greenland and beyond in the high Arctics. And in the Central European lake, you could have up to 10,000 specimens per square meter, and in some highly productive ecosystems you could have up to 200,000 specimens per square meter, which is translated into about 2 kilos of biomass per every square meter, and they're immensely important for facilitating biogeochemical functions, but also for aquatic terrestrial linkages when they transport an organic matter from aquatic ecosystems back to terrestrial. So why chironomids and bioturbation is actually important for the wider context of zoology, ecology, and climate change science is because quite a lot of carbon in the Earth systems is actually buried in the aquatic sediments. We are estimating that about 10% of all sequestered carbon is buried within the freshwater lakes sediments. So the freshwater lakes are only occupying the tiny fraction of the Earth's surface, less than 1%. And if you take away the large body of saline water like Caspian Sea, it's even less. However, this tiny fraction of land surface is burying the same amount of carbon as the whole oceanic shelf altogether. And most of these sediments are populated by animals, most notably chironomid larvae, gammarids, and ephemeroptera larvae mayflies, who are burrowing there. So you could see here the chironomid larvae building the network of the burrows in the lake sediment, and then emerging as adults, dying on the land surface and transporting organic matter. But what is important for us, you see that sediment where chironomids build their burrows changed its color. It's because when animal is burrowing into the sediment, it's changing its density, and oxygen-saturated surface water is going into the sediment pores, bringing in oxygen and nutrients and stimulating bacterial activity. And this stimulation of bacterial activity is changing the rate at which nutrients flow in and out of the sediment, at which respiration is happening, and at which carbon is buried or released back into the water. So this is the basics. The idea about behind the experiments which I conducted in my dissertation was that not only which animals are burrowing is important, but also which kind of sediment is really important. So hydrologists roughly defined in all the sediments in the world water bodies into the two types, based on the type of prevalent transport in it. It's either diffusion-dominated systems or advection-dominated systems. In the diffusion-dominated systems, the sediment is really fine, and the only way for solids and gases to move in it is a diffusion, which is really slow. And advection-dominated systems are made of much more coarse and large sediment particles, think gravel or coarse sand, and their water and gases are flowing through the preferential flow passes, and diffusion is not a limiting factor anymore. What is important here, we think that when animal, say this brittle star, is bioturbating the diffusion-dominated sediment, it's impacting its biogeochemistry a lot because it's creating additional flow and enabling the gases and solids to be transported otherwise than by diffusion, because it's creating this burrow, which is a preferential flow path. And in advective-dominated systems, the burrows doesn't make much difference because there are already a lot of transport passes. So we studied two main experimental systems, lowland lakes in the Central Europe and the marine coastal sediments in the Irish Sea next to Plymouth. And we had two main experimental objects, two model objects, Chironomus group Plumosus larvae or bloodworms and European brittle star Amphura filiformis. So this scheme is just reiterating what I just said, that bioturbation is enhancing the flow of nutrients hypothetically, and we were set to test this hypothesis in the diffusion-dominated systems, which are marked in blue, and probably doesn't make much difference in the red-marked advection-dominated systems. This is a set of experimental hypotheses which were tested in our project, and I would go through them one by one, highlighting what we have learned. And but first I have to stop at the methodology, because methodology is a kind of all the rage in my project, because we were testing quite novel approach. There is a color dye, color tracer called resazurin. It was first purified by an organic chemistry lab of Chicago State University in 1948. And the guys, biochemists there, find out that actually this dye is changing color when it's reduced. And it's reduced in the presence of the mitochondria or aerobic bacteria. And then someone in the food industry decided that actually it's a great test for whether the milk dairy products are still good. So when bacterial titer is low in the food, resazurin doesn't change the color. When it's high, it changes the color, and you probably need to rethink your food standards. And it was used like that to test the vitality of cell cultures and if the milk is still good for about 60 years, until the hydrologist from the Oregon State University in Corvallis, Roy Haggerty, thought, hey, wait a moment, isn't it measuring an aerobic respiration in the system? So he took this dye and he injected it into the stream, particularly into the hyporheic zone of the stream, so the flow underneath the riverbed, which is always called a liver of the river, because most of the biogeochemical processes and detoxifications is going on there. and the group of Dr. Haggerty actually found that you could measure how much oxygen is consumed in the aerobic processes in the whole river using the resazurin. Me and my advisor, Dr. Stefan Krause, sit down together and thought, okay, yeah, well, we could use it if it's only reacting to the aerobic respiration in the system. We could actually learn how much bioturbation is changing the oxygen demands of bacterial communities, and also how much it would impact the carbon sequestration by bacterial communities. So the basic idea, you have a violet-blue dye resazurin, you adding it into the system with the living aerobically respiring cells, and it's reducing to the pink tracer called resorufin, and you could measure the intensity and rate of the process using photometer or fluorometer. And that's how it works into the system. Resazurin is going to the site of bacterial aerobic activity, and then resorufin is appearing back into the water, and we're taking a sample of the water, and we could measure it. And the reduction of the resazurin is very well correlated with the amount of oxygen consumption in the system, with R equals up to 0.98. So this is the mass I figure out behind the transformation rates of resazurin, and basically the amount of aerobic respiration in the system could be described as delta logarithm of resorufin divided by resazurin plus conversion factor of 1, and then there is also a conversion factor epsilon, which describing how many moles of resazurin reduced equals to how many moles of oxygen consumed in the system. So the first experiment was to figure out if we actually could measure amount of aerobic respiration in bioturbated system using resazurin. We were testing two hypotheses, that effects of bioturbation of the oxygen uptake is different into the different types of sediments, and that we actually could use resazurin to prove that. That was our experimental setup. A lot of mesocosms made from IKEA vases, because they're much less expensive than a bromsilicate mesocosms you buy in the scientific equipment shops. With a lake sediment from Lake Müggelsee in Berlin, and different densities of larvae translated into such densities per square meter. And then we injected resazurin, and we measured its reduction for 24 hours. Another thing we done, we placed chironomid larvae into the respirometry mesocosms, sealed with a silicone glue and fitted with an oxygen spot. And we measured at the same time how much oxygen was consumed and how much resazurin was processed, which allowed us to work out the mole by mole ratio and convert reduction rate in our mesocosms into the actual numbers of oxygen consumption. So this is a very interesting graph. I think it's the most interesting results from my PhD. So this regression line is showing the relationships between oxygen consumption and resazurin reduction in the mesocosm with sediment. And you see that there is a good correlation between, and good R-square between the two variables. And this is showing the relationships between chironomid respiration in the empty bottle without sediment and resazurin transformation. And there are actually no significant relationships, which means that chironomids doesn't impact resazurin turnover. First, I was very puzzled with that, and I thought I made some mistake or I screw up experiment, as PhD students do. So I repeated it 3 or 4 times, and the results were the same. And then I said, okay, wait a minute. You are a chironomid biologist, you know a lot about their biology. Think what might be wrong. So chironomids, as many culicomorphan, truly aquatic diptera, doesn't have any stigmas or openings in their tracheal system. They so-called apneustic insects, which means that their tracheal system is never in direct contact with atmosphere. All the oxygen is getting into the system by the cuticular diffusion, which means that water with a dye has no chances to reach in mitochondria of insect and actually being reduced. And that was my eureka moment. So chironomids is changing the respiration of the system, but doesn't impacting the resazurin transformation with their own respiration, which is great, because for the first time in the bioturbation studies, we were able to measure how the burrowing animal is impacting the respiration of the system without actually factoring its own respiration into it. If you just measuring it with a microelectrode, it would measure the whole amount of the oxygen which was consumed by bacteria, insects, inorganic processes like oxidation of pyrites and so forth. But resazurin in this particular system was measuring only aerobic bacterial respiration, which means I was able to measure how much added volume of aerobic respiration bioturbation by chironomids creating. This is the oxygen consumption measured by traditional microelectrode method in the mesocosms with chironomids and without. As you see, consumption in the non-bioturbated mesocosms is linear, and in the bioturbated is exponential, because after some point, when the oxygen concentration is dropping below 25%, chironomids are starting to breathe less because their hemoglobin has a very high oxygen affinity and they're able to adapt to the low oxygen condition. And this is the main results of the experiments. This is a resazurin transformation rates in the mesocosms with different larval densities from 0 to 2112 per square meter. And you see that transformation rate of the resazurin is proportional to the amount of animals in the system. And this is what we figured out for the amount of oxygen consumed. So when we measured oxygen consumed directly with a microelectrode, we found that mesocosms without larvae are consuming 15.6 milligram of oxygen per square meter per day, and mesocosms with 528 larvae per square meter are consuming 29.7 milligrams of oxygen. When we measured the same parameters with resazurin, we got a smaller numbers, actually about 30% smaller, which is great because it's showing that resazurin is actually only measuring bacterial aerobic respiration and discounting the respiration of animals themselves and inorganic processes, and it's allowing us to dissect the metabolism of the sediment for the first time. So the bioirrigation or ventilation of the sediment by chironomids is increased the respiration of sediment to up to 2.5 to 4 times, depending on the amount of animals in the sediment. And resazurin was really sensitive to changes in respiration, and it was explaining up to 69% of the total oxygen consumption when compared to microelectrodes. As I told, because it's actually cutting away non-organic oxygen consumption, respiration of chironomids, and also processes like photorespiration. The second experiment we conducted was posed to learn if the temperature gradient would impact how the animal bioturbation is impacting oxygen consumption in the lake sediments. By the way, all the stuffs are published already and they all open access, so if you need it, I can give you a DOI or a PDF later. So we were testing the hypothesis that impact of chironomid bioturbation in the sediment would change and increase in the gradient of increase in temperature. And this is our results. We have the same mesocosm setup, but only with 3 different density of chironomids, 0, 1, and 2000 per square meter. And we have increasing gradient of temperatures from 5 to 30 degrees at 5 different steps. And as you see, at 5 degrees, there was no significant difference at the impact of different chironomid densities at the respiration. At 10 degrees, there was difference, but it was insignificant. At 15 degrees, there was significant difference between 1 and 2000 on the one hand and 0 on the other. At 20 degrees, we still haven't figured out what happened. But at 30 degrees, there was a staggering difference between uninhabited sediment and sediment which was bioturbated. the bioturbated sediment was respiring about 400% higher or more intense than non-bioturbated. This is really interesting results because, as you understand, 30 degrees water temperature is not normal, or at least it was not normal until the runaway climate change started for the Central European lakes. But now, for example, Tegelsee in Berlin is experiencing about 1 week of 30 degrees temperature per year. During the heat wave of this year, it was even more, as my colleagues told me. Which means that in the bioturbated lake, in the non-bioturbated lake, the respiration is going really low because oxygen solubility is decreasing with the temperature. And, you know, the maximum oxygen concentration in the water is at 4 degrees, and then it's going down from there, and at 30 degrees it could be maximum 7 milligrams per liter, and poor bacteria doesn't have enough for aerobic respiration, so respiration is crashing. But when the chironomid larvae are present, they pump in a lot of additional oxygen into the sediment. And in combination with a high temperature, it's creating a really great conditions for aerobic bacteria, which allowing them to respire, grow, and procreate a lot. And when heterotrophic aerobic bacteria are procreating and respiring a lot, they destroying and mineralizing a lot of organic matter. And mineralizing of organic matter means release of a lot of CO2, which means that normally under the heat wave, lake would mostly shut down its metabolism. But when it's bioturbated, it would actually turn into the source of the CO2, which means high temperature means more respiration, means more CO2, more high temperature. So positive feedback and vicious circle, probably accelerating the climate change. We haven't tested it in situ, but the implications are really interesting. Central Europe has a lot of the eutrophied lakes, or at least alpha mesosaprobic lakes, which has a lot of bioturbators in it. So probably this bioturbators density should be managed under the heat wave conditions in order to reduce amount of organic matter being mineralized. Here you could see the respiration rate, or delta, which is basically the slope of this line changing with the temperature. Oh, sorry. And there was a linear significant relationship between two variables. I mean, this is the most complicated setup of the mesocosms we ever built, and it was breaking down all the time, but we still made it to the publishable results after like probably 20 times failing. Third experiment we did was with marine environment. So we basically done the same thing we done as the first experiment, but with the brittle stars. I went to UK, to the Plymouth Marine Lab, and I was posed to test the hypothesis that aerobic respiration in marine systems would only take a small fraction of the oxygen from the total oxygen consumption, because in marine biogeochemical systems you have a lot of other processes going on. For example, pyrite oxidation, aluminium bauxite oxidation, photorespiration, and other non-aerobic respiration-related processes. So this was our experimental system. We collected brittle stars and sediments in the Plymouth Hoe, which is a big bay next to the Plymouth. And we set up these mesocosms, which were essentially the same as in first experiment, but about 200 times bigger in their volume, because everything is bigger in marine biology. And what we found, we have two types of mesocosms: uninhabited controls and bioturbated mesocosms with animals. And we find out that oxygen saturation was dropping faster in the bioturbated mesocosms, of course, because animals was taking quite a lot of oxygen, but only marginally faster, because it was an advective system with a lot of pores, and as we hypothesized, bioturbated animals are not that important in the coarse sedimentary systems. And this is Resazurin reduction rates. They were higher in the bioturbated mesocosms, but again, only marginally higher. The problem with this experiment, we only were able to repeat it twice, and this is the only one which is still not published, because I don't think we got enough data for publication, and I don't like to publish studies which are not replicated a lot. So I think it's fine to talk about it here as a comparative preliminary study, but you unfortunately, yeah, you could read it actually in my thesis in the supplements, but we haven't published it, and now there is a PhD student in UK who took the mantle on these experiments from me, and hopefully she would get a full-fledged article from that. But what is interesting in these preliminary results, so we studied the diffusion-dominated systems in the freshwater lakes, and in the diffusion-dominated system, the total oxygen consumption has increased by 400%. Aerobic respiration has increased by 400%, and in advective systems with marine animals it increased by about 25%. Then total oxygen consumption, measured not by resasaurin but by microelectrode, is increased by 50% in the lake sediment with really fine grains, and haven't increased at all in marine, which is essentially a confirming the basic falsifying, not falsifying, but confirming basic hypothesis of my thesis that impact of bioturbating animals is really different in coarse and fine sediment. And in fine sediment, like here, it's leading to the skyrocketing aerobic respiration and increased total oxygen consumption, while in coarse sediment, it's just redistributing the respiration, the oxygen consumption sources. It's increasing number of bacteria because animals creating a favorable environment by bringing in nutrients and oxygen, but it doesn't really impact in the sediment all that much. So, I mean, we tested these 4 hypotheses. We were able to fully confirm first, second, and third, and for the hypothesis 4, we need definitely much more experimental data to be confident that bioturbators in the advective environment really redistributing oxygen consumption rather than increasing it. There are several major takeaways. There are several major takeaways. So the controls which are existing on the bioturbation are twofold. There are physical controls and there are biological controls. Biological controls are dependent on what kind of animal is bioturbating. Is it a worm? Is it an insect larvae? Is it a crustacea? And what physiological state it is in. So for example, chironomids during the vegetation season in the summer could, for the Lake Müggelsee in Berlin, the chironomid population there is pumping all the water in the lake, which is about 7 square kilometers in total area, through their burrows within 2 days. So that's quite impressive biogeochemical and hydrological achievement. But in the winter, they're pumping almost nothing. So physiological state of animals is really important. But then species is also important. So we compared only preliminary Tubifex, the oligohead worm bioturbation to chironomids. And Tubifex doesn't change in the oxygen consumption that much because first they have much lower relationship of the body surface to volume, so they need less oxygen. And then chironomids has a highly affine hemoglobin and pumping a lot, so they pump in more water and using more oxygen. But then there are also physical controls, in which sediment animal burrowing and in which physical condition. So is it warm water, is it cold water, is it fine sediment or coarse? All these factors are interplaying and determining the eventual impact of the animal's bioturbation. So another important takeaway is about lake management and how we should change our approach to water governance and lake governance in the changing world under the impact of global climate change. So European Union and other countries around the world has taken major steps to reduce organic pollution of the lakes starting from 1950s, and largely succeeded due to the reduction of the phosphate fertilizers. However, a lot of lakes are still eutrophied and has a lot of organics in the sediment, and they have a lot of potential for the internal eutrophication from this material which is already there. And now when we learn that high temperatures during the heat waves would intensify the biomineralization and release of organics from the sediment, we have to think whether we should do something to control the population of the overabundant bioturbators. Maybe we should, maybe we shouldn't. It's something which should be thoroughly tested, because so many times in history humans were thinking, oh, this animal is harmful, it's doing something wrong, like for example cane toads were introduced to Australia to find the sugarcane beetle, and then everything went south, and now Australia is overrun by Bufo marinus. So I'm not saying that we should be killing bioturbators. I'm saying that we should think in a new way about management of the lake and the nutrients in the lakes, because it's not enough to reduce the amount of fertilizers, because we have a 60 years of history of uncontrolled use of organic fertilizers in the agricultural landscape, and many countries in Eastern Europe, like Ukraine, are still doing it uncontrollably. Ukraine don't have a phosphate pollution threshold. It has one. It has to be high enough to kill the person. Then it's not okay. Otherwise, it's okay. Because, as I told, the rising global temperatures could provoke the positive feedback loop when the nutrients and CO2 would be released from the lakes through the stimulation of the bioturbators' activity. I'm not saying that it's a main way we are getting additional CO2 in the atmosphere. It's definitely global shipping and private cars and meat production. But, you know, it's just another thing to consider. However, I think we could safely forget about it if people would just stop eating so much meat and using private cars. And yeah, the last takeaway, it seems that in the coarse coastal sediment in the sea, bioturbation is much less important for the biogeochemical cyclinite alteration, and it should not be ignored in the biogeochemical models, but we definitely need more data for that. So this is my final take-home figure called Physics Controlling Biology, as a question mark in the air quotes. And I'm plugging back in this lovely brittle star. And with that, I would like to thank you for your attention and would gladly take any questions you might have.
Thank you, Viktor.
Told you 40 minutes.
No problem. Okay, that's great. We have plenty of time for discussion. So, your question?
I have a question. If you also see some effect of the bioturbation on the bioturbators themselves, if somehow they release some kind of dangerous solutions or something, if there could be some connection, like the baseball.
Yes, actually, I didn't talk about that, but if you look at these graphs, you see that with time, that there is a very few difference in the intensity of bioturbation between the treatments containing 1 and 2,000 larvae. This is because we think that mesocosm with 2,000 larvae is pretty densely packed, and they release a lot of metabolites into water. And these metabolites are getting released back into the water, and they could intoxicate animals and decrease their physiological activity. And if you, for example, have heavy metals like cadmium buried in the sediments, they would definitely be released and impact the bioturbation. And we actually, colleagues from Stony Brook University in the New York State has found that for phthalans, well, I guess many of you know what phthalans is, but it's definitely, it's these components, so-called plasticizers, which are added into the plastic bottles together with ibuprofen to stabilize the plastic and not let it to break down. And that's quite a lot of them in the sea, because there is quite a lot of plastic trash in the sea. And they made experiments in the Gulf of Mexico, where there is a lot of plastic trash. And they find out that marine worms are burrowing into the sediments in the major recreation areas where tourists are throwing a lot of bottles and other plastic trash into the sea. And they're releasing phthalens, and the more phthalens they're releasing, the less they burrow. And you introduce in these animals in the mesocosm, and you see that burrows with time becoming less and less deep, because most of the large sensitive animals are dying, and there is a succession in the community, and at the end only animals which are burrowing really close to surface, where phthalens are washed away by surface water, are left.
Question about this climate change scenario, the worst case thing with the never-ending loop of positive...
highly hypothetical.
I assume that chironomids, as many other animals, will have some limits of temperature where they stop to function. So what would be the limits for our species, and are there some warm-loving ones which could then replace them?
So yes, I mean, as I told at the beginning, chironomids could do everything, and there are some worm-loving species. In particular, chironomids are the most worm-tolerated insects in the world. There are some living in the hot springs in New Zealand which tolerate up to 60 degrees, 58, around that mark. But for Chironomus plumosus and other common species, the practical metabolic threshold would be around 33 degrees, after which they would cease to actively function, and after 35 degrees they would die. However, there is a caveat. So Chironomus riparius is a somewhat of emergent model organism. Genome is sequenced, and a lot of people are working with it as alternative Diptera model to supplement the Drosophila. And they found that there are some worm-tolerant races which could function actively up to 39 degrees. And chironomids are very good in adapting to the heat waves because they have a hemoglobin, which is very specific hemoglobin. It has affinity to oxygen about 200 times higher than the human one. And its Bohr curve, this association, dissociation of oxygen with hem ring curve, is very different from ours. So they actually able to tolerate high temperature and low oxygen condition and still bioturbate and bring more oxygen into the water. So actually the red colored chironomids are one of the best candidates to survive in the Central European lakes when the limnophilic mayflies would be gone, limnophilic caddisflies would be gone. Chironomids would be the last higher animals, maybe with exception of tubificid worms, to succumb to eutrophication, hypoxia, and high temperatures.
As far as I know, this hemoglobin is confined only to some chironomids, not the whole family. So can you say that this hemoglobin containing larvae are the most important bioturbators worldwide, or any other species are not that effective?
So the catch is that mostly only red-blooded chironomids are true bioturbators. So indeed, the hemoglobin is confined to about 35% of species in the subfamily Chironomina, and a few genera in subfamily Tanipodina, and to the single genus Propsilocerus in the subfamily Orsocladina. So it's mostly this Chironomina, what is commonly known as bloodworms, what you take with you for the ice fishing or feeding your aquarium fishes with, the most visible ones. They are the most important bioturbators. Other chironomids has different ecological niches. So as Vít could tell you, most of the stream chironomids are very important grazers. Some are shredders or xylophages. Some chironomids are tidal and intertidal marine species. So there are no marine bioturbators among the chironomids, but there are some marine species. And actually, Pontomaya marina is the only true oceanic insect in the world because it's living in the open Pacific. Yes, but I would say that red-blooded chironomids, especially genera Microhieronomus, Hieronomus, Polypedilum, and some Glyptotendipes, are one of the most important global bioturbators, followed by the mayflies from the family Ephemerida and some gammarids. So in freshwater, it's a big three. It's the gammarids, chironomids, and ephemerids. Tubificid worms are also very abundant, but as I told, their mode of bioturbation is not really that impactful. They release a lot of ammonia in the water. That's their main impact.
Can I ask, in Czech Republic, most of water bodies are fish ponds, and with the large common carps, which are also very great bioturbators. So, do you know if chironomids can play any role in bioturbation in such ecosystems?
Yes, they do. I mean, for once, carps as bentophages would bioturbate to get to chironomids. Yeah, I mean, all lakes from which I was taking material are heavily fisheries managed lakes. In Germany, most of the lakes are fisheries managed. In particular, the lake Mugolzea, where I was working, is managed for the Corygonus, the common whitefish, and the eels, and the carps. So yes, chironomids are playing a role there, but bentophagic fishes are sometimes, especially in the ponds, would play a bigger role. Especially if the water body is small and you have a lot of fish, they would be the main sources of the surface bioturbation.
I have idea that maybe high densities of fishes can react these densities of chironomids, so maybe it helps to control. But another question is, if you have any idea why these degrees, about 20, there was different kind of response yes, this graph. Any idea how it works?
Yeah, so I mean the basic idea which we laid out in the paper is that after 25 degrees the solubility of oxygen in the water is dropping drastically, and the thermal stratification is forming in the lake, which is not the case in our mesocosms because it's bubbled. But still the solubility of oxygen is dropping. But chironomids are constantly pumping the water into the sediment, and it's getting more oxygen than by means of diffusion, which is in combination with the high temperature stimulating activity of autoheterotrophic, anaerobic heterotrophic bacteria. That's the main idea we laid out in the publication.
But there is also minimum differences between the densities.
Yeah, I guess because 1 and 2000, it's probably not enough of the difference. Maybe it was not the most, not the best different treatments. Maybe we should go for a bigger difference. But well, yeah. There is a really big difference between so as I explained. After 1000, we see a very little difference between 1 and 2000 because there are a lot of larvae in the mesocosm equivalent to 2000 per square meter. They emit a lot of metabolites and probably they inhibit their own physiological activities by that. And since it's a closed system and metabolites are not going anywhere, probably after a certain density threshold, you don't see much more difference anymore. It's a density, I described it as a density-dependent relationships in the population.
In 30, it surprisingly works.
Let me just ask, what would be the normal densities you get in these eutrophic lakes?
So in Mugolzea, where I was working, the normal density was around 1,200 per square meter. So we took 1,000 as a baseline, something less, but you know, in some
It's
lakes,
not the Masarykova Sea, I mean.
in some lakes like Mewbottom, maybe some of you heard about it. It's in Iceland. Densities are going up to 200,000 per square meter, and I mean, in the lakes across the board, let's take your average lake in Germany, Czech Republic, Austria. It would be 500 to 1,000 of large larvae and some smaller larvae of the other species. So you would have about 1,000 large Chironomus and then some other stuff.
Then I have a question. I wondered a bit about this comparison of the marine system with the brittle star, and the limbic system.
You mean that one?
Yeah, with the limbic system with chironomids. I mean, of course, these animals look very differently. I mean, there's a huge size difference, but also, I mean, there are probably other differences. Now,
yeah,
one of the pictures you showed of the chironomid larva, it looked like they have this U-shaped type of tunnel. So wouldn't it be better to use the budworm, the Orenicola marina, for example, which has a very similar shape? It's much bigger again, but if you don't have any marine chironomids doing the job, then maybe this would be a better equivalent?
We thought so. I wanted to go with Orenicola marina or Nereis diversicolor when I came, and I talked to marine biologists, and I came back here because you see this classification of type of sedimentary working. It depends on the burrow. So chironomids and brittle stars by extension are biodiffusers, which means they moving the sediment from the burrow sideways. And Orenicola marina and Nereis worms are upward conveyors. They push in the sediment upwards. So actually, the brittle star also has essentially a U-shaped burrow. Water is going from here to here. This is a housekeeping chamber. So this arm doesn't ventilate, and this arm is used to move the food particles to the mouth. So essentially, I was pretty surprised, but marine biologists told me that that would be the closest functional analog of chironomid you could find. You know, the analogies are not always straightforward. But yeah, I mean, when I came, I thought, oh, I want to work with Orenicola marina. Yeah, and you couldn't compare it.
Okay, thank you. Then I have one comment. You said that Darwin's book, the last book, basically, on the origin of vegetable mold did not get much attention. Well, I'm a soil zoologist. I'm working with annelids. So
at the moment of publishing.
That's exactly, okay, we will have to go back to literature, because what I heard on conferences is that actually it was a bestseller and many more volumes were sold than of this original species. At his time, I heard, maybe you said it was already after his death, so maybe it was 10 years later, but so I'm not sure about this information.
Okay, I would not argue. I mean,
but I'm not sure, you know.
I mean, that's what I read in one review paper, and I would not claim that I'm an expert on the matter.
I heard exactly otherwise.
I heard that it was underrated, and you know.
It's definitely underrated, because earthworms are underrated.
Yeah, but yeah, thank you for your opinion. I would strive to improve my knowledge regarding how it was received by public.
I'm not sure if I got it correctly. In the introduction to your talk, you mentioned that in the lakes there is actually more carbon dioxide stored than in the seas.
No, not really. What I was trying to tell is that there is a comparable amount of carbon is sequestered in all of the world lakes to the continental shelves. In the sea, it's definitely much more carbon. But I mean, continental shelves has much bigger surface areas than the lakes. I guess about 60% of all carbon is a blue carbon, so it's in the sea. I don't think that lakes are sequestering more than 8 to 10%. I think that the main difference is that sequestration is in the lakes used to be more permanent, because it was just going into the anoxic sediments and stayed there. And I mean, So lakes are considered the second most important source of semi-permanent carbon sequestration after the bogs and swamps, because the sphagnum swamps are sequestering carbon pretty permanently, unless, you know, some stupid government decided to dry them up, because it sounds great.
Okay, thank you, Victor. There are no other questions. I think it was exhaustive and exhausting discussion.
From the word ex-hauster.
So thank you, Victor, once more.